What's up, YouTubers? Here in this last video that I'm going to make, we're going through how to run the bioinformatics tool hmmmer. The previous videos showed how to install and download and install those programs, and now we're going to do a quick run through of using hmmmer to look for a specific sequence in a large genomic data set. So as in the previous video, we have our target sequence and we went to NCBI and other genomic databases and we downloaded those sequences. And so the result here is this TEF1 ref fasta file. So for my in my specific scenario, I'm looking for a gene within a genome. The gene is called the TEF1 alpha gene, also known as the translation elongation factor 1. And so I've collected a large number of these sequences from NCBI, and you can see in my FASTA file here, if you do a head, this is one of the sequences. We can go ahead and look at the whole file here. So this is organism 1, this is a fungus Armillaria gallica, this is the TEF1 sequence from that organism. And then these are different samples of the same species, and then as you can see, you start to get into other organisms. Like here we have another fungus. So anyways, we can count how many sequences are in this file with this pretty complex command here. It's kind of like a control F. So it counted how many of these symbols are in this FASTA file. So there's 1,856 sequences within this file. And when you're creating your FASTA file to search for your genes or a specific genome sequence, one thing that you want to keep in mind is that you want to have a broad spectrum of diversity from the sequence you're looking for. So in my example, I was looking for I'm looking for the TEF1 alpha gene in this organism here, Coniferporia sulfuracans. So as part of NCBI, I went and I downloaded any TEF1 genes that I could find from that organism. I also looked for other closely related species, such as example Coniferporia warei, Phlinus noxus, Fomitoporia mediterraneae. I downloaded the TEF1 sequences from all these different organisms, and then I concatenated them into this one file called the TEF1 ref fasta file. So after I have my fasta file, what I'm going to do is I'm going to create a multi-sequence alignment, and for this I'm going to use a program called mafft. So for this command I'm running mafft. I'm going to use my FASTA file, and then I'm going to create a multi-sequence alignment. So we can see here it's creating that file. I'm going to pause this because it might take a minute. Alright, so then our multi-sequence alignment file has been created, and we can go ahead and open that up and see what that looks like also. You can also use, there's many other viewers that you can use to view this offline on your desktop. But it essentially just aligns up the sequences so that they match consistently with all the other sequences. So then from there we're going to use hmmmer, and we're going to build a .hmm file within hmmmer. So for that we use this command here, hmmbuild. This is the name of the .hmm file we want it to be called, and this is our freshly made multi-sequence alignment file. So we do that. It should be pretty quick. That's one convenient thing about hmmmer. It's very fast to check and make sure that it actually works. You can do ls-lh, which shows you the file size. So now that we see that there's it's more than 0, we know that something has occurred within that file. After we build a .hmm file, now we're going to copy over a full genome sequence that we want to test. So this is a full genome sequence for a Coniferporia sulfuracans sample. And if we do head on that, we can see the contents. It's just different nucleotides, A, C, T's, and G's. There should be around about 39 million of these. So what we're trying to do is look for a gene, the TEF1 alpha gene, that is about 1,200 base pairs, and we're trying to find that from a file that contains 39 million base pairs. So to go through this manually, it would just be ridiculous and would take forever. So that's why we're using hmmmer, which is this really great program that allows us to search through these genomes really quickly and extract the sequence that we're looking for. So we've made our .hmm file. We have our spades or our scaffolds.fasta file, which is our full genome sequence. So then you might see this hmmsearch. This is what I was trying to use originally. You have hmmsearch, your freshly made .hmm file, and then your test file. And when you run this, you get this error where the target sequence length is 100K, greater than 100K, and as I mentioned, this file is around 39 million base pairs, and so this hmmsearch cannot search through files that are greater than 100,000 base pairs. So for that, if you get this error, then we're going to use this other built-in function within hmmmer called nhmmer, and then we're going to do our hmmmer file, that we made, and then our genome file. For that, we're searching. It's now searching through the genome and looking to match up to see if there's any matches with the TEF1 alpha gene based off of the file that we use that contains over 1,800 different gene sequences. And so you can see that we have some hits here, and the output is a bit convoluted and kind of complex. So to get past this, we're going to do another trick here, which this is a one-liner script here that we will do the same search, but this is going to output all of the output into a nice new FASTA file for us. And this is using this program esl-reformat. So this is our search here like we did, and then this symbol here is the pipeline. So along with our hmmsearch, we're also going to ask this program to write out the data into a FASTA file for us. And so when we run that, let's see if it works here. And if everything worked as it was supposed to, okay, so it didn't, because you can see there's 0 in there. So we're going to try this again. We'll break it down just one step at a time here. So we're going to do the search, but we're going to have it output the information from our search into this a-2.hits.txt file. takes a minute. hmmmer is pretty quick overall. So then let's see if we have, we do have some data within the a-2.hits.txt file. So then as a final step here, we're going to use that ESL format to write out the results from hmmmer into a nice simple FASTA file for us. Oops. You get that error a lot in Linux. Just wants to make sure you don't overwrite a file that you might need. And so now if we look here in our a-2.hits.fasta file, you can see that these are our TEF1 alpha gene sequences that we extracted from the entire full genome using hmmmer. It's a really great tool. I hope this tutorial is really helpful. Please feel free to like and subscribe to this video, or leave a message in the comments of other videos you'd like me to make, and I'll try to make them. This is a way that helps me to learn, and I hope helps other people to learn. Alright, so thanks for watching, and have a great one.